Our next speaker is, we move on to flies now. Our next speaker is Li Zhao. She's from David Begun's lab in UC Davis.
Good evening. Firstly, I would like to thank the organizers for giving me the opportunity to talk about my recent work. As an evolutionary biologist, I'm interested in understanding the patterns of evolutionary trajectories, which include divergent evolution, convergent evolution, and parallel evolution. Ever since Darwin time, convergent evolution has been treated as one of the most important evidences for adaptation. There are many nice examples for convergent evolution, especially related with morphology and behaviors. One of the very nice examples is that multiple lizard species are able to independently evolve into similar color pattern to adapt to the local white sand or grass environment. Another very nice example is that the African cichlid species are able to independently evolve into similar color pattern to adapt to local environment. So far, most of the evidence about convergent evolution are related to morphology and behavior. Now, genomics has provided us a tool so that we are able to understand what's the molecular genetic basis of convergent and parallel evolution. So we are able to understand the convergent evolution on the DNA level, RNA level, and pathway level. In Drosophila melanogaster, the species I'm interested in the last couple years, we already know that there is a nice parallelism for the East Coast North American cline and East Coast Australia cline. Although there are evidences supporting that demographic history might play an important role for this pattern, it is also worth knowing that natural selection is playing a very important role for such pattern. I am very interested in studying Drosophila melanogaster and Drosophila similans. So basically, I wanted to compare the patterns between species. These two species are very similar. They have a lot of similarities. They both have very large population size. They are cosmopolitan species that are also human commensals. The divergent time between Drosophila melanogaster and Drosophila similans are only around 2 to 3 million years. So they share recent common ancestors. At the same time, they have a lot of differences as well. Compared to Drosophila similans, Drosophila melanogaster has much greater geographic and clinal variation. And it seems like there are lots of controversial studies, but it seems like both species, they are responding to temperature in a different way. And on the DNA level, Drosophila similans has no common chromosome inversion, while for Drosophila melanogaster, inversions, especially inversions on chromosome 3R, is very important for local adaptation and geographic variation. So I decided to use gene expression as a phenotype to study if these two species share parallelism. So the questions I'm interested to understand is, what's the magnitude of geographic clinal expression differences between these two species? Do the two species exhibit parallel expression adaptation? Do the two species exhibit parallel expression plasticity? In order to answer this question, we firstly collected a lot of Drosophila melanogaster and Drosophila similans flies from nature. So we collected flies from Maine and Panama. One of them is in the North American temperate region. Another one is in the North American tropical region. So for each isofamil line in each species, in each population, we raised them at 21 degrees C and 29 degrees C with 3 biological replicates. After that, we did the RNA-seq and analyzed the data. Firstly, we want to understand what's the difference for within species. So the first question we want to understand what's the geographic expression differences. So we compared between the expression between Panama and Maine. We found that we found that for both species, there are quite a few of genes that are showing differential expression. You see that these differentially expressed genes generally accounting 66% of total genes. And the thing is, one of the interesting things is that although Drosophila similans tend to have less geographic variation on the DNA level, on the expression level, the variation is as much as Drosophila similans, and actually at the 21 degrees C, they even have more geographic expression differences. In terms of gene expression, chromosome distribution, for Drosophila similans, there is no bias. But for Drosophila melanogaster, chromosome 3R is enriched with the geographically differentially expressed genes. So inversion 3RP is also playing a very important role for this pattern. Then we studied the magnitude and directions of the gene expression. We found that the magnitude and directions are was significantly greater at the 29 degrees C. So for example, here is the pattern of the 29 degrees C. One of the interesting things is for Drosophila melanogaster, there are many more genes showing higher expression in Maine population, and for similans, the pattern is exactly opposite, suggesting that these two species might have different ways, or at least a part of genes that are different to adapt to the high temperature. If we check about the 21 degrees C, there is no such pattern. For us, the most interesting question is if these two species share parallelism for the gene expression. So we used the one-to-one oslogs between Drosophila melanogaster and Drosophila similans. And we found that for both 29 and 21 degrees C, there are around threefold enrichment of shared genes, suggesting that there is strong parallelism for the gene expression variation along the line. We also checked about the expression direction. So what I mean direction is if a gene tends to express highly in Maine or highly in Panama. We found that for the shared genes, most of the genes, they are having the same pattern in Drosophila melanogaster and Drosophila similans, supporting that those patterns might reflect the true biology. Then the next question is, what's the biology in it? Since we used RNA-se to do this experiment, we checked about the geo-enrichment at first. For Drosophila melanogaster, there are a lot of significant geo-enrichment, among which the ones I'm very interested in is that at the high temperature at 29 degrees C, there are a lot of genes that are apparently associated with phototransduction and reduction pathway. It suggests that maybe detection of light is very important for Drosophila melanogaster to adapt to the local environment. For Drosophila similans, we did a parallel analysis. Except these two marginally significant terms, we found nothing. At first, we were wondering if it's because of the annotation quality. We then used the homologous genes of Drosophila melanogaster, and we tried to do an enrichment, and the pattern still holds. So it seems like Drosophila melanogaster is, it seems like Drosophila similans has no, the response to geographic variation is rather generic, but not related with specific pathways. There are lots of studies about plasticity, because plasticity is very important for adapting to local environment. So in the past decades, there are a lot of nice examples about phenotypic plasticity. However, it's not very known that if geographic variation in expression plasticity adaptive. Since for our experiments, we raised all the flies at 21 and 29 degrees C, so that we are able to use temperature as an environment factor to study plasticity. So firstly, we want to compare within species. We found that for both species, there is greater expression plasticity in Panama, which is a tropical population for both species. So this is opposite with a classic view that a lot of people think that in Drosophila species, temperature population tend to have strong phenotypic variation. We then compared between species. So for each population, we compared the ones between Drosophila melanogaster and Drosophila similans. We found that there are 12 to 42 genes that are shared between species, suggesting that temperature plasticity is generally very conserved between the species. We then want to understand if geographic differences in plasticity has parallelism. So in order to understand this, we studied the genes that have plasticity in Maine or Panama population, but not in the both population. Then we use these genes to determine if these genes tend to show population-specific plasticity. And the answer is yes, there are indeed significant enrichment, suggesting that geographic differences in plasticity exhibit some level of significance as well, significant parallelism as well. So adding together this slide and the previous slide, we can see that there is a component of plasticity variation that is highly conserved, and another component showing evidence of recent parallel local adaptation. The last question we want to understand is the G/E effect, because gene and environment interaction is very important for maintaining maintaining natural variations. And this is a classic reaction norm for gene and environment interaction, showing that the fitness effect of alleles differ between the environments. So we did the same analysis in both melanogaster and similis species. And we found that there are more than 200 genes in Drosophila and melanogaster showing gene-environment interaction. However, in similis, there are only 7 of these genes showing G/E, suggesting that these two species might have different way to maintain the natural variation. So with this, we have our conclusions. We found that the two species exhibit significant geographic parallelism for gene expression differences. The two species exhibit significant geographic parallelism for gene expression plasticity. We found geo-enrichment patterns are very different between these two species. The genomic extent of G/E interaction was much more common in Drosophila and melanogaster, suggesting that these two species are having different mechanisms for the maintenance of natural variation. With this, I would like to thank my advisor, David Began. He's a great mentor and a wonderful scientist. I also would like to thank my co-authors, Nicholas and Yannicka, as well as my colleagues at UC Davis. I'm contributing 2 posters, and I would like to speak with you if you are interested. And at the end, I want to mention that I'm going to move to Rockefeller University. If you know somebody that might be interested in our research, please ask them to contact me. Thank you. Yeah, sure.
Have you thought about just repeating this work with just, say, 2 random populations at just 2 different geographic areas, regardless of north-south, because there'd still be genotype-environment interaction, I guess?
Yes, it's a good question. So since for this experiment, we used a lot of isofamil lines. So basically, for each data point, we have around 15 to 30 isofamil lines. So we think that this pattern probably would be pretty robust for at least for this endpoint. We now also have some data that is in the, not in the endpoint, but in some other populations. Seems like this pattern still holds, but we don't know what's the true biological reason for this yet. So it would be interesting to study 2 random population that is not associated with Klein. Yeah.
Any more questions? So we'll move on to the next talk. Thank you again very much.
Oh, thanks.